Usually people thank the organizers, but that seems strange for me to do. Thanks for coming, Brian. So maybe I'll thank the sponsors. So thanks to CIRM, Sanford, AbbVie. Really, really appreciate you helping to make this possible. Let me get oriented here. So as Lineage is a publicly traded company, I can refer everyone to our filings and safe harbor clauses, more information at sec.gov. I'm going to talk today. It's a very strange presentation for me because I'm going to speak only about OPC1, which is our lead program for spinal cord injury. Before I do that, I just want to orient you to the company. We're a local company. We're right up the road here in Carlsbad. We have administrative offices and a new lab space in Carlsbad, and we also have a manufacturing facility, a GMP manufacturing facility located in Israel. About 78 employees overall. So we're not a big company, but we're also not a small company. We're somewhere in between. All right, so what we are prominently tasked with is the manufacture and replacement or transplant of specific cell types of the body. So we make, colloquially speaking, replacement parts for patients. So we have 5 programs, 5 different cell types that we're manufacturing today. And one of the things that we do is we harness the power of self-replicating pluripotent cells, or what a lot of people will call stem cells. The nice thing about those cells is they provide an unlimited source of starting material because, to just be technical for a moment, these cells don't have a hay-flick limit. So you can culture them forever and they can be an unlimited supply of material, but they're undifferentiated. They have to become something else before they go into the patient. And that is a lot of our intellectual property, a lot of our technology is knowing the recipe for how to take an undifferentiated cell that hasn't decided which of the 200 cell types of the body it's going to become, and we give it the instructions. We say, okay, we want you to do this, turn left, turn right, and only become an oligodendrocyte, or only become an RPE cell, or whatever it is that we are aiming to make to then treat the patient. We've had some validation of this approach. It's a little bit of a different approach. It shares some similarity with some others, but we're really proud that our ophthalmology program actually was the subject of a nearly $700 million license agreement with Roche and Genentech. So the whole notion or the concept of using a differentiated cell to replace and restore function in a patient has rewarded us in part with a $50 million upfront payment just for something we're doing in the eye. So we're hoping for, some parallels here, this a halo effect with what we're doing in the spinal cord. All right, so there are more steps than just the 2, but just to kind of give you a sense, we start with these unlimited pluripotent cells. We don't do any genetic editing. We just give them the normal, natural, biological, developmental biological instructions that they require to become the oligodendrocyte progenitor cell, which is our target cell. We never put residual stem cells, residual undifferentiated stem cells into a human being. And this is a one-time treatment. Our notion here is that if we can get the cells in there and they remain in the patient, they're not rejected, and if they are functionally active, that they may help an individual regain additional motor function. So these are the 5 programs, but again, I'm only going to focus on the spinal cord injury program today, which additionally has received significant financial support over the years from CIRM. But I'm actually going to tell the story in 2 sections. The first section, many of you know better than I do. I've only been the CEO of Lineage for about 4.5 or almost 5 years, but the program predates me tremendously by at least that long, if not longer. It actually started as an asset at Geron and was some of the founding technology for Geron, which itself was a very, and maybe continues to be a famous company in the space. Geron elected to pursue their oncology program, unfortunately to the detriment of the spinal cord program. And after treating just 5 patients in a thoracic setting, they moved on. And the assets found themselves in a small company called Asterias. Asterias did some work. They additionally treated 25 cervical patients and found some really evocative phase 1 data that I'll show you in just a couple of slides. But they ran into some challenges, and we ended up buying that company. We ended up buying Asterias. So I'll get to that. That'll be part 2, but I'll get to that in a minute. But what essentially is happening here is the manufacture and the use of an oligodendrocyte progenitor to reestablish the myelin sheath around the axons to help promote signaling, so that nerves that wire together fire together. Many of us learned this way back when. This is our idea for how you might be able to increase functional activity after an injury by providing the very cells with the very kinds of content material that are required for effective and efficient signaling along the pathways. These are allogeneic, so we're not taking cells from a patient and manipulating them, putting them back in, and that's because we're treating in a subacute setting. There's no time to do that. You have to have, for this particular approach, you have to have an off-the-shelf or an allogeneic option. So that's nice because it means it's the same material for every individual. The other question we often get when you're putting cells in people is, aren't they rejected? Aren't they quickly rejected? So our protocol calls for just 60 days of immunosuppression. So you don't have lifetime immunosuppression with this therapy. It's relatively short. What happens when those oligodendrocytes get inside of the individual? Factually, nobody ever really knows, but these are the three things that are probably very prominently involved. So the myelination of the axons, secretion of neurotrophic factors, and then reducing the cavitation. Electrical impulses have a heck of a time jumping across gaps, so you got to have some kind of substrate in there. So these are three things. It would be very difficult for me to weight these and tell you what's most important. Other work we've done might suggest that the neurotrophic factors are the least critical, but that's in the eye. I don't know if that's really applicable in the setting of spinal cord injury or not, but these are three things that we think are really important with our approach. So what happens when you transplant these cells into individuals in a subacute setting, 3 to 6 weeks post-injury? Well, we're encouraged, right? This is a single-arm study, so it's very difficult to have absolute statements from it. But what we see suggests to us that there is a signal here, a beneficial signal. Almost everyone on this trial improved. And there was only one individual who didn't fall into that category. And when we go back into some of the larger databases and we try and find matched controls that look like the patients that we enrolled to kind of create a synthetic comparator arm, we're pretty happy with what we're seeing, because we think our patients, I guess I really should say the Asterias patients, did better when treated with the OPC1 drug candidate, product candidate rather than what you might have expected in comparison. And so this certainly for us merits further development and answering the ultimate question, which is how much benefit, if any, how much benefit do you get? And that's going to require a larger study and a control arm in some way. But we're very happy to see that, that this clinical data was generated. The safety is, of course, an important consideration. Notable that there were many hundreds of adverse events associated with this trial, but only one of them was potentially associated with OPC1. It was a grade 2 dysesthesia. It auto-resolved. The reason why I pay attention to this slide and why I think this is important is, as you've heard throughout the day, ultimately the FDA's responsibility or authority is to render a decision as to the risk and benefit. And so if you have a therapy for which the risk is very low, it's very well tolerated, you don't have a lot of side effects and complexities, it would suggest naturally that the amount of benefit that you need to demonstrate to offset the risk can be small. Now, that in itself is encouraging. However, it runs afoul of the fact, fortunately, that there aren't many, many millions of people with this condition, right? So when you're trying to show small effects and show them at a level of significance and conviction, one way you solve that problem is by putting more and more patients on study. It's really hard to do with an orphan condition. So, the solution is probably better assessments, which is one of the themes of this conference. How can we measure things that matter more to the patient and get alignment with FDA on that? And how can we measure things more sensitively so that we are not using blunt tools to detect smaller benefits? So the connection of the clinical data from the phase 1 study that was conducted and the safety profile, I think, is really encouraging. We also, perhaps not too surprisingly, saw a massive reduction in the cavitation because we are putting material in there. These are living functional cells. They can divide, they can occupy a space. We think this is a very favorable attribute of the therapeutic. And we're pretty happy to see that these implants have been extremely durable, extremely well tolerated. There have been no reports of people, getting cancer or growing kidneys where they're supposed to have spinal cord. Like all the crazy stuff that you see in sort of sensational headlines from decades ago is just not happening in our hands or other hands that I'm aware of. Jake, are you happy with your cells? They're still in you somewhere, right? Yeah, okay, yep, thank you. So, isn't that crazy? I mean, it's years ago. They're still in there, right? Maybe doing good things. All right. So these studies have been published recently. They're both published in 22 in peer-reviewed journals, so they're available for you. The top-line takeaways, as stated here, are, again, directionally encouraging, very well tolerated, people generally doing better. But the ultimate test is, remains to be a conclusive and, presumably large and extremely well-run study in order to answer the question, how much benefit do you have? Can you rely on that? And should many, many people who suffer from an SCI receive this therapy? Okay, so how are we going to do that? Chapter 2. So I think it was only like 2 or 3 months after I joined Lineage that one of the first sort of press releases or headlines was we said we were going to acquire Asterius. And that is because we have some really world-class manufacturing capabilities. And one of the challenges with this program is that the cells that were being manufactured weren't quite up to where we thought they needed to be commercially viable and to really pass muster with FDA and to become a commercially available product. So how do you move from fascinating and exciting phase 1 data to, no, it's an actual product. You're going to want this out there and useful and easy to use and so forth. And part of that is in the manufacturing. In fact, we have an entire sort of campaign around, and I talk often about what makes a successful cell therapy, because you can't just put it in and get an effect and be like, all right, I'm good, I'm done. You really have to be able to master all of these categories, the reproducibility, the control of your program. So we've been focusing on all these red things that I just highlighted, the scale of production, right? It has to be economically feasible. Those are the things that we worked on. So we acquired Asterius, and then this program kind of went quiet. And for those of you who literally were involved in this program for many years, you might have wondered, like, what happened? It went into a Lineage and it went dark, right? Nothing is happening. Oh my gosh, a lot was happening. We were trying to solve these problems so you can have an actual product, not just a publication. Potency I left in green on purpose because the new material that we're manufacturing and how we do it now has retained the potency, so I don't worry about that, right? I think the potency is there, but it's these other areas that we wanted to emphasize. So we have proudly now a very long list of changes, favorable changes to the original process of manufacturing these cells that move it from interesting and promising to, no, it's real. We created all sorts of new analytical methods. We retained all of the potency. We increased the scale. We diminished the presence of undesirable cell types. A lot of this generated new intellectual property for us. We got rid of animal reagents. I mean, it really was, thankfully, because it was sort of this bet, like, can we fix these things or not? We've been successful in fixing these things. There is one slide in particular that I really like to show from that bullet list, and it's this one. And it's a little nerdy, but this is a scientific, kind of environment, so it's okay for me to do that. But I love this slide because these markers down here that are anonymized, those are surface markers of undesirable cell types. So this is essentially a scale of impurities. And the gray material that was originally used, that is what it looked like. And you can see upwards of 20% undesirable cell types, or at least the presence of surface markers associated with undesirable cell types, right? So, i.e. shorthand, impurities. The orange stuff that doesn't really even get to 2%, that's what we're making today. So I really like this because it's an illustration of Lineage's expertise in manufacturing, but beyond that, it actually reflects a maturation of the field. We're not the only ones working on glial cell types for spinal cord injury. And all of us who are working in this area with this particular approach have benefited from much better and more powerful reagents, much more sensitive analytical tools. The field is maturing, like it or not, and of course we like it, in a way that's making this more feasible. So you don't have to put, junky stuff into people. You can actually put stuff that you truly have a very high level of control and reproducibility and scale for the economics of it, et cetera. So this is the stuff that we want to use going forward. There was another area that we needed to fix, which was on the delivery. So the original way that patients were treated was there's this large gantry or scaffold that is attached to the patient's bed, and it is all there to hold the needle. And it was a giant pain in the buttocks. It had a ton of parts. There was training to put it together, and guess what? Not every bed is the same size, and so you're always kind of adjusting. It's fine for a phase 1, totally fine for getting some initial data. But if you want to roll this out into a large trial with 30, 40, 50 centers, or you want to have this be a commercial approach, this isn't going to cut it. And one of the key deficiencies of this approach is that you had to disconnect the patient from the respirator while you were administering the cells, right? So it's like tick, 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 right? That's not a great way of doing it. So we've been working on a new approach, which is utilizing a much more clever and well-designed device that actually attaches directly to the patient. So the wonderful thing about that approach is that you do not need to have the patient disconnected from the respirator when you're administering the cells. Chest cavity is going up and down. There's a floating cannula that travels with. You can put the cells in much slower than the old way. So I like that. That's safety, right? It also has far fewer components. Everything that's in the surgical field is a lot tighter and nicer. And so we're really excited. This will be the next thing that we do in addition to the cells and the purity and the things that we fix there. This is the new device. We've already talked with FDA about this, and they said that we can also use this in chronic patients for the first time. That'll be really exciting. And then a third thing that we worked on is the formulation, and that is because the old way of delivering these cells required the cells to be shipped to the site, washed, plated, counted, handled, manipulated, hopefully not dropped and lost. That's all gone. We came up with a brand new patented formulation where the cells are already in a frozen state. They're just thawed. About 5 minutes later, they're going into the patient. So we've eliminated 99% of all of the dose preparation activities. So we really are professionalizing this program so that if it does show benefit in a larger and convincing trial, it's going to actually be a product someday and have a much faster route to get there. So some of the takeaways from this is a very old study that's been done, but there are a lot of learnings, a lot of experience. We're going to apply those lessons to what we're doing. The next clinical study is going to be focused on the device, but we're also going to get those cells going. It is now time for this program to reemerge, and conveniently it's happening in connection with this conference because it's no longer hiding in the back, trying to figure out how we're going to deliver it, how are we going to make the cells, what are we going to do. We've solved these problems, and now we're heading soon to get back into clinical testing and bringing this very promising program back. So I like this hashtag replace and restore because it kind of captures everything that we do. I do think that there, of course, is an important place for small molecules and antibodies and stim and all of that. But one of the things that I like about whole cells is that you're covering a lot of things, right? If you are replacing the entirety of the cell, you get to go beyond just a single pathway. You're really addressing a lot of things. So it may ultimately be an approach that works best in combination, and we've been having conversations even today about how we might explore that, and how it can work in conjunction with physical therapy, stim. We don't know, but we're really excited about trying to answer some of these questions. So thank you very much. As always, the patients are inspiration, and I'll be here all day if you've got any questions. So thank you.